and this summer I've had the honour of working at the biochemistry department of the University of Otago doing a studentship. I'd like to thank the Otago Medical Research Foundation and my sponsor Fiordland Discovery for funding this project and making it able to happen. So my project involves me looking at an RNA and trying to find out whether it leads to colorectal cancer.
The central dogma of genetics is like baking a cake. The DNA is the recipe book. The RNA are the ingredients you need to make the cake. And the proteins are the cake made at the end. However, not all ingredients need to be made into a cake to be eaten. For example, you'd more likely just eat the chocolate than making a cake every time. This is the same in biology. Not all RNAs are made into proteins. A large majority, which we call non-coding RNAs, are functional by themselves.
For my project, I am working with a non-coding RNA that has been found to contribute to breast cancer, and I'm testing to see if it also contributes to colorectal cancer. And in order to do this, I used a genetic technique called CRISPR interference.
In the DNA in a cell, there is the gene of interest that I am studying. Without interfering, this gene will be made into non-coding RNA. However, with the CRISPR interference technology, this can be stopped or interfered with. There is a complex of proteins that contains a repressor protein whose role is to stop genes being made into RNA, and there's also a guide sequence, which brings the whole complex to the gene of interest like a magnet. When the complex is bound, RNA can no longer be made.
By putting this CRISPR interference complex into colorectal cancer cells, I'll be able to see if there is decreased cancer growth and progression when I take away the non-coding RNA. The aim of this project is to be able to determine whether this non-coding RNA does contribute to colorectal cancer, and if it does, it'll open a lot of doors for drug development in the future. And the aim is to help lower the mortality rate of colorectal cancer, which is the second biggest killer in terms of cancers in New Zealand.